we can move on to Ricardo. It's my great pleasure, as you might imagine, to introduce Ricardo, who will end this conference. And obviously, it's very special for me to introduce Ricardo because I worked with him for 5 years in London. He's a mentor and, importantly, a friend. And today he's going to talk to us about what his lab has been doing to push the boundaries of super resolution, and importantly, all underlined by open technology. The floor is yours, Ricardo.
Thank you, Pedro. As you might realize, I'm also equally proud to have you as one of the key organizers of SPALM, and to be close to you. So thank you, everyone, for coming to my talk. I'm going to dominantly focus on telling you about some of the open tools we have developed in our lab, or that we have helped collaborators establish. So the key point I want you to keep in your mind as you look through this talk is that everything that you're going to see is immediately available to you. You can just go out of your lab, and likely, if it's software, it's immediately open for your use. If it's hardware or probes, it's very easy to replicate them. And since I had a talk just before Martin, if you see some cool movies in my talk, they will likely be inspired by Martin, who has done fantastic movies illustrating his work. So, as you might realize, I've rejoined the Portuguese and Spanish microscopy community in the last months. I'm currently moving my lab back to Lisbon. So I'm quite proud to be closer to you now, and I very much look forward to interact more and more over time. So as you realize, we have developed quite a bit of an amphitheater of tools for microscopy. And during this talk, you already heard a little bit about some of them, such as, for example, nanoscale fluidics presented by Pedro and Roman, which was partially developed when Pedro was in our lab, and Roman's presenting zero-cost deep learning for microscopy. We're also quite proud to have contributed a little bit to the development of CARE that Martin has presented, and to the use of nuclear pores as reference structures in supersolution microscopy, which Jonas Breeze has presented also. And because of this, I'm going to exclude these tools. I'm going to dominantly focus on what you're seeing here in orange. All right, I always like to start by telling you a little bit how I came into this. I'm actually a little bit lucky because I know exactly which moments as a kid did I decide to become a scientist. And it all boils down to this experiment that you're seeing here. I was brought into a laser physics lab and simply asked, when I was 15, what do you think will happen when you shine a laser through a slit? And my thoughts at the time were, okay, so laser beam, there's a slit. I knew a few things, although I was a kid. So I knew light was composed by photons. I thought of them as ping pong balls with a certain momentum. And there's an aperture there. So what I thought was, okay, so some ping poles are going to cross the aperture and some will collide against the aperture. The ones that collide either get reflected or absorbed or something happens to them. So my hypothesis was that I get a shorter beam at the end. So we did the experiment, and here's a movie of it. And what you're going to see actually blew my mind. It was crazy, right? Because what you're seeing here is exactly the opposite to what I was expecting. As you close down that slit, that light actually broadens and starts getting these side lobes. And this was one of the key moments in my life where I was presented with something that I couldn't explain at all. And because of this, right there, I kind of decided to spend my entire life studying this process. And if you think about it, what you actually have on a microscope is a point source of light, to in a large degree, very similar to what you have with a laser, that has to cross an aperture. The difference is now is that that aperture is now circular instead of being that 1D slit. And because this aperture is circular, the fact that you have at the end is the same broadening that you saw before, except instead of being in a single dimension, it's completely radio asymmetric, right? But it's exactly the same process that you had at the end. And it's this diffraction effect that happens whenever you have an aperture that ends up defining the resolution limits that you have at the end. Every point source of light becomes broadened up, and you cannot distinguish those points of light anymore. And the way that everyone's using single molecule causation microscopy supersolution, the field that I like to work on, has found a way to deal with this is by decreasing information content. Instead of looking at samples like this, where you have all the fructifers actively emitting light, you create a time event where you're able to switch off most of your fructifers and turn on a very small subset. Because this small subset is really simple, there's less information there, you can actually detect and localize those fructifers. And if you cycle through turning off and on fructifers, you get to detect and reconstruct a representation of the fructifer sets switched during your acquisition. A little bit as you've seen Jonas presenting before. So this is what I got in as a PhD student. And it was fun because I remember starting to build microscopes to do this and looking to novel probes that were able to create these blinking events. I did that while I was in Paris. And one night I was invited to go to a picnic in front of the Eiffel Tower, and I saw this. And the funny thing about it is that I was really annoyed when I saw it because I was already spending 8 hours in front of a microscope trying to get cells to blink. And I didn't really want to see anything blinking in my free time. However, this is really beautiful because it tells you about how physics is universal. My mobile phone that is collecting this movie that you're seeing here, it has a lens with a circular aperture, exactly the same way that my microscope has a lens with a circular aperture. So the same physics that are imposed in that aperture on the microscope are physics that are imposed into the aperture that you have on my mobile phone. So that means that exactly what I'm filming here is exactly the same thing that I'm trying to film on the microscope. So back then, I was developing an algorithm to do single molecule coalization called QuickBound. And we could actually apply it to the Eiffel Tower because it's exactly the same thing that you're seeing on the single molecule causation microscopy experiment. And thus, you could actually super resolve the Eiffel Tower. Okay, so then a few years back, I was invited to start at a lab in London. And this is what I wanted to do. I was an expert in supersolution microscopy, so I wanted to use supersolution microscopy to really study small supermicro complexities in cells that could not be easily resolved by computational microscopes. So viruses, vesicles, host-pathogen interactions dominantly. And I'm going to give you an example of why supersolution is cool to address some of these questions. I'm going to show you, for example, here, the deadliest virus we ever faced as mankind or humankind, pox. It killed millions of people. It's actually kind of responsible for the reason why Europeans colonized America, because pox decimated the native population when the Europeans went there. But it's also quite a big virus, which means that even with structural illumination microscopy, which just increases resolution by a factor of 2, you can already start getting structure. And one of the beautiful things about viruses is that extracellular viruses have a tendency to have a metastable structure. That means that you have, for example, some of these viruses in the cover slip, you're going to see the same structure coming over and over and again, just in different rotations. And because of this, one of the things that we created was a software called VirusMapper. And VirusMapper pretty much just tries to find these rotations and orientations to align all these viruses in order to create a structural map of the final structure that you have in 3D. And with this, if you actually do dual labeling with fluorescence, you can always use the reference structure that is set to then map some other structure against it, which means that we can start building a map of tens of different proteins and where they actually are physically within the virus itself. with quite a structure. To get the best of both worlds. Right. So this is what I wanted to do once I started. But here's some of the feedback I had with can you guys hear me? The computer is having effect. Go. Okay.
Go ahead.
So, sorry. I'm just going to move on to here. And you can see my screen, correct?
Right.
Just double checking. Okay. So, I was telling you, sorry about that interruption, by the way. Started the lab, wrote some first grants, and here are some of the feedback that we had. From Human Frontiers, this would be a great project for cryo-EM. From the MRC, Supersolution in general should be seen at face value as a sophisticated way of analyzing blobs that can only be complementary to high-resolution EM. So I was obviously annoyed with some of this feedback, but there is some grounds of truth in here also, which is what have we seen with Supersolution that we have not seen by electron microscopy before? And the answer is there's very few things.
Ricardo, I think
So,
that we cannot see your screen.
You cannot see your screen.
Okay, hold on. Can you see my screen now?
Yes. Okay. Now
I was saying reviewers say Supersolution sucks, and there is some claim of truth there, which is the fact that there hasn't been that many structures we have discovered with Supersolution that we had not seen by EM before. So why is Supersolution interesting? One of the reasons why it's interesting at the today. Okay. One of the things that is clearly interesting about Supersolution is the capacity to do microscopy, which is not possible with electron microscopy. This means that you can actually have the chance to look at dynamics.
Okay, so while we wait for Ricardo to come back, we'll just take this opportunity to ask a few of the questions that were left hanging for Martin. And to start, curiously enough, it's a question from Ricardo. So maybe we'll skip that one. But there's
my answer.
From Bran and great work, Martin. A question about StarDist plus classification. Can it use information from multiple channels? Example, cells with a Fucci construct?
I assume multiple channel as an input, or just because so I assume, so basically the input of StarDist can be any channel, any number of channels. So the output would be an instant mask and a probability, class probability per cell. So if you want to base the classification and the instant segmentation on all the multiple channels, then this is possible. The only thing that is not possible is, and I think there was a thread in the forum recently, to base, like if you have a 3-channel or 2-channel image, and you only want to classify the outlines of the object based on one channel and the class of the objects based on the other channel. This is currently not possible. So you cannot switch, you cannot dedicate a specific channel, although this in principle is not, that might be feasible to do, but we just haven't implemented it correctly. But if your input is multi-channel and you want to use all the channel for both classification and segmentation, that's no problem.
Okay, yeah, so Bran basically answered in the chat saying that yes has an input, red and green fluorescent channels.
Oh, yeah. I don't see the chat correctly, or that's in Hoppy probably.
Yeah,
I am hopefully back.
okay. So, we have Ricardo back, so let's try to carry on with the presentation, and then we'll go back to Martin in the end and meet the speaker to answer. There's still a few more questions left. Okay, thank you.
Yep.
The floor is yours.
Thank you, Pedro. I'm actually going to bypass things a little bit since I killed some time, and I apologize. I don't know what happened with the network. Hopefully it's stable now. So I wanted to tell you a little bit how we got into SRRF, but many of you might already know a little bit about it. So I'll go directly and tell you about some of the new things we've been working on. So we've developed a lot of software in the lab and some hardware with Pumpy, for example. And one of the things we wanted to do is try to also improve the way that probes work in supersolution imaging, particularly in single molecule microscopy. So for this, we actually started playing with hairpins as a potential probe for supersolution imaging. How does this work? Well, in practice, what we've done is we grabbed microbeacons, which are these hairpin probes where you have a loop side and a small self-hybridizing stem. And on 3' ends, we put a quencher, and on 5' we put in a fluorophore. When these two are close to each other, they actually don't emit because there is collisional quenching between these two moieties. But if the probe actually opens up, you get the quencher to be far away from the fluorophore, and thus the fluorophore will start emitting light. The interesting thing here is that you can design the sequence for these probes in a way that at room temperature, they are unstable, which means that they're constantly switching between the closed state, where they don't emit because the quencher is next to the fluorophore, and an open state where the fluorophore is away from the quencher. And this is great because in reality, you get the blinking that you want with single molecule quantitative microscopy, but the blinking is generated dominantly and mechanistically with this thermal unbalance on the structure of the sample, instead of being generated by, for example, pumping fluorophores into a triplet state, which requires very strong illumination. So we wanted to put tests, and we actually designed these probes so that we could bind them to a strapped evidence encoded cover slip. And here's how they actually look. What I'm going to do now is I'm going to add formamide, which is going to force the probes to open up. Thus, you're going to switch between that blinking state to a constantly emitting state, where you can actually realize where probes are actually bound to the cover slip. So formamide now. There. Good. So in practice, here's a way that we can generate photo switching that is dominantly independent of the illumination that you put into the sample, because it's this opening and closing mechanism that is generating the photo switching. And just to give you here some time traces of 3 of these probes that we've selected, you see that beautiful blinking before, and upon adding formamide, they just constantly emit for you to know that there are probes actually there. Now, the interesting thing here is that because these probes just naturally blink, and they even blink if they're not being illuminated, one of the cool things here is that we don't need strong illumination, and we do not need special buffers to induce that blinking. So in principle, they are more life cell compatible than probes that you would use for this term. However, they are synthetic. So one of the big problems we are still facing is how do we get them inside cells, and how do we get them to bind to proteins of interest? And thus far, the way that we've done that is actually by using antibodies, which means we're still mostly looking at fixed and premiumized samples. But in the future, we'll be looking into ways to actually directly using click chemistry with them inside cells. All right, and one of the bits of the last stories I wanted to tell you. You had beautiful talks about image analysis and machine learning throughout this conference. But there are certain tasks that our brain is still beautifully designed to do, particularly if those tasks involve teaching a machine learning algorithm how to do things like classification or segmentation. And to help in this, Stephen Lee's lab is the lead in this work, but to help this, we developed a new VR system which allows you to explore single molecule concision microscopy data. And the beautiful thing here is that our brain is actually great at making sense of what that point list in the volume represents, and therefore segment structures or even teach machine learning algorithms how to segment structures from that point data. All right, and I'm sorry I didn't get a chance to present you a big chunk of the talk, but I wanted to tell you and to finish with this, which is, this is our eye sucks. It sucks because it's to a large degree poorly designed. We have our vasculature in front of our optical nerve, which means that there is a series of elements that actually distort our vision. And we even have a blind spot in our retina, which prevents our visions to a certain region of our field of view. However, you don't notice this. And you don't notice this because your brain is quite good at correcting all those anomalies that our eyes have. And here's the beauty of evolution, right? That unfortunately, there's not going to be 1 or 2 gene mutations that are going to solve the architecture of our eye. We're probably stuck with this indefinitely as a species. But the plasticity of our brain is actually able to compute all these problems and correct for them. And if you have doubts about it, get drunk. If you get drunk, you actually are going to sense a lot of the mechanisms that your brain has to correct for these anomalies, and they're going to actually be taken down as you get happy with the alcohol. But it's also a really nice time in microscopy because the same thing is happening. There hasn't been really disruptive changes in the way that we use the physics of glass to look at samples. I could argue this a little bit, and there's some beautiful work done by Andrew York, for example, in trying to improve the way that we design objectives. But it's not major changes to that architecture at the end of the day. And the revolution now is actually on the way that, given those limitations that we have on the physics of glass, we're able to extract more and more data of it by digital analysis. So it's a really cool moment in time because of this. And to wrap up things, if you're an undergraduate student and you're looking for a PhD, we have open applications for the optical biology PhD program at UCL. It's a great program if you want to both play with biology and learn about advanced optical techniques. So please do apply. And I am thankful for you for listening and everyone that helped us in some of this work. So happy to take any questions you may have.
Hey, Ricardo, thank you so much for the talk, despite the connection challenges. Now it seems all sorted. So I can start with a question regarding your talk. So what plans do you have to develop next? What is your next challenge now that you move to Portugal?
So good question, Pedro. I can hear you. So one of the things that we're obviously interested in is adaptive microscopy. As in, we now have the capacity to improve images in real time and also classify the content of those images. This means that we can actually have microscopes that react to the behavior of samples. And this is really important because there's some really cool experiments that can be done, particularly in host-patient interactions, where you have certain key events in time that are transient, and you want to explore those specific events. So that's very much the direction that we're trying to go. Obviously, as with everything else we do, it's meant to be reproducible and transferable. So everything that we're going to do is going to be completely open source. So not only to discover new things in biology, but also to make those discoveries transferable to other problems that people might want to explore.
Lovely. Okay, so we are about on time, and we still don't have questions. People are saving themselves for the Meet the Speakers session, I presume. So maybe what we'll do is thank you, the speakers, for lovely talks, and end the plenary session of SPAWM, and then we will move to Meet the Speakers just after this. Thank you so much.
Thank you.